All right, and on to lecture 2 for day 10, where we'll finish up the lower GI. Here we'll be focusing on those accessory digestive organs, and we'll talk about the digestion that occurs within the lumen, and then we'll move on to the large intestine. And this will be the last lecture that I give before Clark takes over. And that means it's also your last worksheet. And your last set of memes. Science memes, I suppose. Forget 2020, like you forgot the Krebs cycle. That was a online submission. And this one, I can feel this one. Whoever stole the purple marker, please return it. No. First few days of last semester, the markers would be stolen by the end of every day. So that was interesting. Thankfully, a student brought me a whole set. And finally, we got this one. I thought it was hilarious. All right, let's move on. We'll start with a general summary of hepatopancreatic secretions, and then we'll differentiate between endocrine and exocrine function of the pancreas. We're going to take a small step back here and return to the accessory digestive organs when we look at the digestion that occurs within the lumen of the small intestine. So the accessory digestive organs secrete bile and pancreatic juice. Bile is produced by the liver, stored and concentrated in the gallbladder, and then we have the pancreas that secretes pancreatic juice, which consists of digestive enzymes. In the last lecture, we looked at digestion within the brush border, how those peptide fragments and disaccharides are broken down into the individual amino acids and monosaccharides, respectively. Today we're going to look at the initial steps that occur within the lumen of the small intestine. And these will include total fat digestion, protein being broken down into peptide fragments, and carbohydrates to saccharides. We'll begin by looking at the pancreas, and that sits both behind and below the stomach. So if I were to superimpose our stomach over that, it would look something like that. A little bit below and then behind. And the pancreas secretes both endocrine and exocrine products. Endocrine products are going to move to the blood, while exocrine are going to be secreted into the lumen, which is technically exterior to the body. And these are going to be the digestive enzymes that we are interested in. Next, we have the liver. And the liver sits directly beneath the diaphragm and has many functions. They include excretion of bilirubin, cholesterol, hormones, and drugs. It's involved in the metabolism of fats, proteins, and carbohydrates. Enzymes are activated within the liver. It's also the storage of glycogen, vitamins, and minerals. We see the synthesis of plasma proteins like albumin we've talked about before and different clotting factors. And then we see it's involved with blood detoxification and purification. However, today we're going to focus on its role in the production of bile salts, and these are needed for the emulsification of fats. Last thing I want to point out is this hepatopancreatic ampulla. So we break that down. Hepatic means related to the liver, and pancreatic, pancreas obviously, and then ampulla, and that's a small cavity. So what this point is, it's where the pancreatic duct joins the bile duct from the liver. So we see that secretions from both the pancreas and the liver pass through this ampulla before they enter the duodenum. All right, now we're going to talk specifically about the pancreas. Here's another view, and you can see the stomach outline here that would be in front of it. It's this elongated gland. We mentioned the pancreas has both endocrine and exocrine function. Just a quick touch on the endocrine function. We have these clustering, and they're shown here in violet, these clusters of pancreatic cells. And those cells, termed the islets of Langerhans, they secrete insulin and glucagon. You've heard of these probably before. Insulin allows the cells to absorb glucose from the blood, while glucagon triggers a release of glucose from the liver. While those endocrine products, the hormones insulin and glucagon, enter the blood, so here we can see a release from those islets and Langerhans, and it'll move into systemic circulation. We're going to be focused on the exocrine products of the pancreas. So here we can see these grape-like clusters of cells, and these are called acini or acini. And if we zoom in on one of these, we can see they consist of these acinar cells. And these secrete the digestive enzymes. Those move through the duct, we can see here, and then they empty into the duodenum. We also said that bicarbonate is secreted simultaneously from these accessory organs to help neutralize the acidic chyme that enters the duodenum. So we can see here within this acini, we have these duct cells. And the duct cells secrete bicarbonate that is also going to move into the duodenum. And all those products are going to enter the duodenum through that hepatopancreatic ampulla or duct. We'll now look at the different pancreatic enzymes, and we'll also look at whether they are initially active or they need some type of activation to function. One of the astonishing things about the pancreatic enzymes is that they can almost entirely digest material within the lumen all by themselves via the secretions of the acinar cells. Now those acinar cells are going to secrete proteolytic enzymes, so proteoprotein, lytic breaking, so they're going to break down proteins, breaking things like peptide bonds. We're going to continue to break down carbohydrates. So we had amylase within the oral cavity. Here we have pancreatic amylase to continue the digestion of carbohydrates. And then we have pancreatic lipase. Lipase breaks down fats. And then we'll continue to break down a little more via the emulsification process. An important characteristic of all three of these secretions is that they perform optimally within an alkaline environment. So we saw the simultaneous release of bicarbonate from the accessory digestive organs along with those secretions. So that's going to help neutralize the acidic chyme from the material that's coming from the stomach, and it's also going to create that environment that these secretions need to function optimally. We'll start by delving a little deeper into these proteolytic enzymes. The first proteolytic enzyme we will discuss is trypsinogen. This is an inactive form of trypsin. This is similar to the pepsinogen pepsin mechanism that started protein digestion within the stomach. Now we're going to continue the breakdown of those proteins. So trypsinogen, I'll highlight it down here, is going to be converted to active trypsin by enteropeptidase. That's an enzyme found within the brush border. And then active trypsin can hydrolyze peptide bonds to break down proteins, and that active trypsin will also activate other enzymes, as we'll see shortly. After trypsin is activated by enteropeptidase within the brush border, it can convert the other proteolytic enzymes to their active forms. So we can see here that we also have chymotrypsinogen, and then via trypsin activation, this will be converted to active form chymotrypsin. We also have procarboxypeptidase, and that'll be converted again by trypsin to carboxypeptidase. Now, these enzymes attack different peptide linkages. So this is going to give more thorough digestion of proteins within the lumen. We're going to be left with both individual amino acids after this digestion, as well as small peptide fragments. And then we then saw how those remaining peptide fragments were digested completely within the brush border at the end of the last lecture. Next is pancreatic lipase. Okay. Now this one is extremely important because it's the only enzyme secreted that can completely digest fat. So this occurs within the duodenum, and here we'll break down triglycerides into monoglycerides and free fatty acids, and these can readily be absorbed. Now this enzyme is also secreted in an active form, like pancreatic amylase. Doesn't need activation. It's not going to harm the pancreas. For a think, pair, share, what would occur if you had a deficiency in these pancreatic digestive enzymes? How would that present itself clinically? Well, it may present as steatorrhea, and that is the presence of undigested fats floating in the feces. Now, if we have pancreatic insufficiency, we'll have other sources of enzymes to help digest proteins and carbohydrates, right? We have amylase within the saliva. We can start digestion of proteins within the stomach. But lipase only comes from the pancreas. And the pancreas is almost solely responsible for the digestion of fat. So we'll see that floating undigested fat within the stool. Now we'll look at the regulation of pancreatic secretions and how they're related to cholecystokinin and secretin. Got a nice crying, screaming kid in the background for you there. Good ambiance. When are these pancreatic juices then secreted? So we'll now turn to the regulation of the secretions. We will see the release of pancreatic secretions in response to chyme from the stomach entering the duodenum. And the release of the secretions is under control of hormones, and specifically the enterogastrones. And these include secretin and cholecystokinin, CCK we'll call it. When chyme enters the duodenum, the acid stimulates the release of the hormone secretin. And secretin is released from the duodenum from secretory cells there, and it's going to travel in the blood, as hormones do, and ultimately reach the pancreas. Upon reaching the pancreas, it's going to stimulate the release of sodium bicarbonate ion, and that's going to help neutralize the acids within the duodenum, creating that optimum pH environment that we talked about earlier for proper enzymatic function. The other enterogastrone, cholecystokinin, or CCK, is going to be stimulated and released by the presence of fat. So here we're going to get CCK released from endocrine cells within the mucosa of the duodenum of the small intestine. When that's released into the blood, it's going to travel again to the pancreas. Here it's going to stimulate the release of digestive enzymes from those acinar cells. Those enzymes, including lipase, will be released. I say including lipase because this relies on a negative feedback system. So this is stimulated by the presence of fat, but as we release our digestive enzymes, including lipase, we'll decrease the amount of fat there. With decreased fat presence within the duodenum, we'll get less release of CCK and then less release of the digestive secretions from the pancreas. Quick check for understanding. Let's say we have acids and undigested fats that are detected within the lumen of the duodenum. Which hormones will be released? A, secretin, B, CCK, C, both, D, or none. If you said both, you are correct. So the acids within the duodenum, that'll lead to the release of secretin, and secretin will lead to the release of bicarbonate from the pancreas. Presence of undigested fats will lead to the release of CCK from the duodenum, which will travel to the pancreas via blood and lead to the release of all the other digestive secretions from the pancreas. In case you missed that last question, we'll do a quick review. You can pull out your worksheet here if you've printed it off. So we'll begin with the presence of acid in the duodenum. Okay. That is going to lead to secretin release from the duodenal mucosa. There's cells there that are going to release secretin. That secretin, being a hormone, is going to travel in the blood to the pancreas, where it acts upon the pancreatic duct cells. And remember, those duct cells secrete sodium bicarbonate. And then we can see here that is going to work to neutralize that acid within the duodenum. If there's fat within the duodenum, we'll see the release of cholecystokinin from endocrine cells in the mucosa. That hormone will travel again to the pancreas, where now we're going to get release from the pancreatic acinar cells. And they're going to release our digestive enzymes into the duodenum. Those are going to go back, and one of those digestive enzymes, remember, was lipase, so we'll decrease the amount of fat and via negative feedback, shut this system down. Now we'll look at the functional anatomy of a liver lobule, including the actions of the hepatocytes and the Kupffer cells. The liver is our largest metabolic organ, and we're going to be talking about its role in the secretion of bile salts. These will help with both fat digestion and absorption. All right, real quickly, I want to give you a little bit of background on the liver here, relating to how it receives blood from two different sources. So we'll see that oxygenated blood, leaving the aorta, will flow into the hepatic artery. Hepatic, again, meaning liver. So this is how oxygenated blood is going to be brought to the liver. Okay? But we'll also see the hepatic portal vein. And this is where venous blood from the digestive tract is going to enter the liver for processing and storage of absorbed nutrients that we've gotten from that GI tract. And then we'll see there's a hepatic vein that leaves the liver, and that'll flow up to the inferior vena cava. The liver is made up of 4 lobes. So if we're looking at it from the front, we can see there is a right lobe and a left lobe. Then if we flip it and get a posterior view, we can see additionally there is a caudate lobe and a quadrate lobe. Not too concerned that you know the lobes of the liver, but kind of an FYI. Now, each lobe of the liver contains many different lobules. It's about 100,000 lobules in the right and left lobes. And lobules have this columnar hexagonal pattern to them. And that consists of hepatocytes, or liver cells, and vessels. And they're all oriented around a central vein. So here, if we zoom in on one of these lobules, we can see those individual hepatocytes that make up the lobule. We can see the vessels, and then we can see that central vein. Now, the hepatocytes are responsible for all of the metabolic functions of the liver except one that we'll describe shortly. Incoming blood will enter a lobule via a branch of the hepatic portal vein. And remember that incoming blood from the hepatic portal vein, that's the blood that's coming from the GI tract that is nutrient rich, although it is deoxygenated. So that's going to be delivered to the liver for processing. And upon entering the lobule, the blood is going to undergo that processing. So we'll see storage of nutrients and detoxification of blood as well. Some of these events are going to occur within these hepatic sinusoids. So we can see these violet areas of the lobule that are lined with these liver cells, the hepatocytes. Now, the exchange of nutrients is going to occur between the hepatocytes that line these sinusoids. So the sinusoids allow a place for the blood to go where they can exchange those nutrients with the hepatocytes, the liver cells. But then we can also see within these cells, albeit not very well, we have these Kupffer cells. Okay? And these Kupffer cells are involved in phagocytosis. So they're found within those sinusoids where the blood travels, and they're going to remove old red blood cells and particulates, including bacteria. So we see the hepatocytes, while they do most liver function, getting things out of that blood, for example, the phagocytic activity is going to occur within the Kupffer cells, within those sinusoids. Here we can see that characteristic hexagonal pattern of a liver lobule. And we can also see that at each point of this hexagon, there is a set of 3 different vessels. The first is the hepatic artery, and that is shown in red. So that's going to deliver oxygenated blood to that portal system. And then we have in blue the hepatic portal vein. So this is going to be that deoxygenated but nutrient-rich blood. And then we also have shown in orange, that is the bile duct. So this is how bile is going to move away from the liver lobule. So while blood will be moving toward the lobule in the hepatic portal vein and artery, the bile is going to be moving in an opposite direction, out from the liver lobule. Here we can see the vasculature of a lobule a little bit better. So we'll have blood entering from the hepatic portal veins, and as it works its way through the lobule, it'll be processed by the hepatocytes and the sinusoids, or those Kupffer cells, and it'll end up within this central vein. Also, the oxygenated blood from the hepatic artery will move into the central vein. And all the central veins, they're going to converge to form the hepatic vein, which carries blood away from the liver. Now, the digestive function of the liver is the production of bile. And recall that we said that the hepatocytes, the liver cells, they can carry out almost all the liver functions by themselves, and that includes the production of bile. The hepatocytes continuously produce and secrete bile into these small channels here called bile canaliculi. So the bile flows through those from the hepatocytes till ultimately it hits the bile duct at the periphery of the lobule. That bile duct will then ultimately converge to form the common bile duct. We'll now spend a little bit of time on bile. We're going to look at how it's formed, the components of bile, when it's released, how it's recycled, and then the action of bile as an emulsifier. While bile is continuously being secreted by the liver, it only enters the duodenum during meals. The sphincter of Oddi, shown right here, guards the opening of the bile duct into the duodenum. So between meals, that sphincter is going to be closed. And if it's closed, the bile is redirected to the gallbladder. It's not shown here, but it is here, this green structure, until it is stored until it's released when another meal occurs. What is bile? Well, while it is not pretty, it does consist of bile salts, cholesterol, lecithin, and bilirubin. Lecithin works as an emulsifier, and bilirubin, that breaks down heme. So when you break down red blood cells, you're left with heme. Bilirubin can break that heme group down. Now, we're going to focus on the bile salt component of the bile. Now, they're a derivative of cholesterol, but they're involved in taking large globules of fat and turning them into a liquid emulsion, a process we'll work through shortly. And after participation in fat digestion and absorption, most bile salts are reabsorbed within the blood. And that's part of the reason we said the ilium is so long within the small intestine. That's to allow for proper reabsorption of these bile salts. Unfortunately, precipitates of the bile can form stones within the gallbladder called gallstones. And they form from bile cholesterol and bilirubin if your levels are too high. Those stones will form in the gallbladder. And the remedy is, well, not a remedy, I guess you can either pass these stones, which is painful, or the gallbladder can be removed without much side effect. The bile duct, shown here, will actually expand a bit to hold bile. So, and it's enough that you can adequately emulsify fats with a meal. What are the cues for bile release? How is the release of bile regulated, in other words? There are two mechanisms. They are hormonal and neural. The hormonal mechanism involves cholecystokinin. And remember, that's released when fat enters the duodenum. Now, the cholecystokinin is going to cause contraction of the gallbladder to release the bile. Simultaneously, it's going to cause relaxation of the sphincter of Oddi. And the sphincter of Oddi is what allows the bile to enter into the duodenum. The neural mechanism, on the other hand, leads to bile release when the vagal nerve stimulates secretion when we begin eating. So this is similar to that cephalic phase where we taste, smell, think, or see food. It's going to trigger a parasympathetic response leading to bile secretion. And finally, 95% of bile salts are reabsorbed within the ilium. So we take a look at this figure here on the right. We're going to have bile salt release from the liver, and it'll enter the GI tract where it'll flow through. And then at the lower section of the small intestine, the ilium, we'll see that 95% of that bile salt is going to re-enter the hepatic portal vein system. And remember that hepatic portal vein is then going to go up to the liver where it can deliver the deoxygenated blood that's carrying the nutrients, but it's also going to have 95% of these bile salts ultimately. Now we do lose 5% of bile salts that do not get reabsorbed within the ileum, and they move on to the large intestine and they're lost in the feces. But most of it is recycled. This is called the enterohepatic circulation. Entero related to the intestine, hepatic, liver. So we see intestine, liver circulation of the bile salts. Shown here is the molecular structure of a bile salt. We can see there is a hydrophobic nonpolar end, and that's represented by this cholesterol group here. Remember that bile salts are a derivative of cholesterol. And then we can also see that there is a hydrophilic polar region. You can see this negative charge here associated with this polar region. Okay. So what happens then with the molecular structure is we end up with a negatively charged water-soluble portion. The polar thing, they've flipped the orientation on us a little bit here. So we have the negatively charged polar side. That's going to interact with things like water very well. And then we have this lipid-soluble portion due to that cholesterol, or the hydrophobic nonpolar side. The hydrophilic and hydrophobic properties of the bile salt are going to be important for emulsification. This will allow for adsorption at the surface of a lipid droplet. So adsorption basically means it's going to form a thin film at the surface of the lipid droplet. So this lipid droplet is going to be nonpolar because it's a lipid. And then it's going to be surrounded by an aqueous solution that has water, which is going to be polar, H2O. And we have both lipid soluble nonpolar side to this bile salt, but we also have a hydrophilic polar side that's negatively charged. So these bile salts are going to come along, and that lipid soluble side is going to plop onto that lipid droplet, and then the polar water soluble side is going to interact with the aqueous environment surrounding it. And in this manner, they're going to form this thin film of lipid salt at the surface of the lipid droplet. We'll see that those bile salts absorb to the surface of the lipid. Let's say we have a large droplet of fat. Well, then contractions of the intestine are going to help break that up into smaller little droplets. And when that happens, the bile salts adsorb to the surface of those smaller liquid drops. Now, because the nonpolar side of that bile salt orients to the middle and adsorbs to the surface of the lipid droplet, that means the negatively charged polar area of that bile salt orients outward and interacts with the water. Now, because there's a negative charge associated with that polar side of the bile salt, ultimately these repel each other due to those negative charges being oriented on the outside. It creates this shell of negative charge, and these droplets repel one another. And we call this emulsification. And this is key because it prevents these molecules from rejoining into a larger fat droplet. And it also increases the surface area for pancreatic lipase to then break down this liquid. When we have a large fat droplet, relatively, there's not a lot of surface area exposed, but when we break them into these smaller droplets, we increase the surface area by a lot, allowing lipase to get in there and do its job. Real quickly, we'll look at fat digestion and absorption. All right, so let's look at how that fat is then absorbed. We'll start at the top with a large droplet of fat, and this is going to consist of triglycerides. We'll see that through the action of bile salts, we can emulsify that droplet. So we've increased the surface area exposure then to lipase. So lipase will come in and further reduce them to monoglycerides and free fatty acids. This is where the other components of bile come into play. So lecithin and cholesterol. They're going to combine with bile salts to form these little spheres or micelles. Now, micelles will have monoglycerides or the free fatty acids at the center of them. And the micelles are able to fuse with the plasma membrane and move into the cell. Now, here's the weird part. Upon entering the cell, those monoglycerides and the free fatty acids reform into triglycerides. And here is where they're packaged into chylomicrons, which can travel undisturbed through the central lacteal and into the lymph. And the last thing we'll do with lower GI is look at the basic anatomy, motility, and defecation reflex of the colon. Seems like a good place to stop. We entered through the mouth, and now we're exiting the anus, and that's the end of it all. The last section of the small intestine, the ilium, joins the large intestine at the cecum. That's the first part of the large intestine. They're joined at the ileocecal valve. So ilio for ilium, cecal for cecum. And this valve is very important because it prevents the bacteria-laden contents of the large intestine from contaminating the small intestine. The small intestine has really nutrient-rich content, and the bacteria would multiply rapidly. So this is under control. This valve is under control, the ileocecal sphincter. Now, with a new meal, it's going to push that valve open and relax the sphincter as material moves through. Once in the cecum, it pushes the valve closed and contracts the sphincter. Here we have the sections of the colon. These are pretty straightforward in terms of structure names. They're kind of just named accordingly. So we'll begin with the cecum, which we just worked through. And then we have the ascending colon, because it ascends. We have the transverse colon, because transverse means across. Then we have the descending colon, sigmoid. Sigmoid, of course, means S-shaped. This is your sigmoid colon. And then the rectum, and the rectum means straight. And then we end with the anus. The material that enters the large intestine from the small intestine consists of things that were undigestible, such as cellulose. We can't break that down. Or unabsorbed biliary components. So we said 5% of the bile salts aren't reabsorbed within the ileum. Well, then those are going to pass into the colon, the large intestine. And then we'll also have the remaining fluids, importantly. So the colon is going to work to extract those fluids, water, and our salts, and it's going to dry out and compact the contents to form feces, which will eliminate through the anus. So the primary function of the large intestine is to store the feces before defecation. If you've ever drawn large intestine, or intestines in general, you tend to draw them like this, right? With these sacs or pouches. Well, unknowingly, you are drawing haustra. And those are the pouch or sacs that occur within the colon. And they occur because we have these rings of smooth muscle. And when they contract, they'll form a little pouch in between, or haustra. And those contractions will shift every 30 minutes or so and give rise to new haustra in new location, new sacs or pouches. And these are not propulsive contractions. They kind of shuffle the material back and forth to expose it to the mucosal layer to increase absorption. These haustral contractions are the primary type of motility that we see within the colon. And just like small intestine, they're initiated by pacemaker cells that give rise to this basal electrical rhythm. If haustral contractions aren't propulsive, how then do we drive the feces forward to defecate? Well, this is a product of mass movements, which are how they sound. Essentially, after a meal, so after each time you eat, maybe about 3 or 4 times a day, you experience massive contractions. And this is where you get the ascending and transverse colon to contract simultaneously. And that's going to drive the feces into the distal part of the large intestine where the material is stored until you can defecate. The gastrocolic reflex is what initiates the mass movements after a meal. Now, it's mediated by gastrin. Remember, gastrin is released by G cells within the stomach. So when food hits the stomach, gastrin's released, and that's going to kick off this gastrocolic reflex along with parasympathetic innervation. You'll probably notice that you get these mass movements. They're most noticeable right after your first meal of the day when you get that urge to defecate. And that'll bring us to the defecation reflex, which instead of defining here, we'll work through on the following slide. All right, last slide and last part of your worksheet, and we'll look at the defecation reflex. So that's going to be initiated when the contents of the large intestine are driven forward by mass movements. That's going to stimulate stretch receptors within the rectal wall, or baroreceptors. That distension information is going to be sent via sensory neurons to the spinal cord. And then via parasympathetic motor neurons, that signal about the additional distension is going to increase nerve signals to the smooth muscle of both the sigmoid colon and the rectum to give rise to contraction, while the inner anal sphincter is simultaneously relaxed. But thankfully, the external anal sphincter is under conscious control by our cortex. So at which point, if we decide, we can then contract our diaphragm and abdominal muscles to assist in that defecation while simultaneously relaxing that external anal sphincter.